Hi, everybody. I'm Chris Doherty, Vice President for Academic Affairs and Dean of the Faculty at Chestnut Hill College, and I'm delighted to be joined this afternoon. We're having this conversation on the afternoon of Thursday the 19th by Dr. Kenneth Soprano. Dr. Soprano is Professor of Biology at Chestnut Hill College. He holds his PhD and Master's in Microbiology from Rutgers University, a bachelor's degree magna cum laude from College of the Holy Cross. He's been professor at Chestnut Hill College since 2008. Ken served as vice president for academic affairs and dean of the faculty from 2008 to 2012. Prior to joining Chestnut Hill, Dr. Soprano was vice president for research and graduate studies at Temple University, where he also served as acting vice provost for research, as professor, as director of the program for graduate studies in molecular biology and genetics at the Fels Institute for Cancer Research and Molecular Biology, also at Temple, in addition to his faculty role and his extensive publications in his area of expertise. Ken, I'm delighted that you're joining us for this conversation around everything we're seeing with regard to the COVID-19 phenomenon. I just reminded those who are joining us that we are speaking on the afternoon of Thursday, March 19th. What are you seeing happening today? What can you share with us in terms of your observations?
Thanks, Chris. I appreciate you asking me to participate in the program and for having the opportunity to have this discussion. I think that, obviously things are changing every day, and we're getting reports every day of new cases, and unfortunately, in some cases, deaths as well. I think today's, perhaps most striking piece of information came out of the CDC report, which has looked at the numbers of Americans that have been hospitalized as a result of the infection, and surprising realization that 40% of those who have been hospitalized are actually young people. They're between the ages of 20 and 50. Of course, everybody is aware that the virus seems to be much more fatal for the elderly. If you're over 65, the amount of fatalities is substantially higher, especially if you have other complications. So that, every day there's, seems to be one new thing that comes out, and in many cases, these are somewhat surprising pieces of information. And I think that in and of itself is surprising. And the sort of take-home message there is that young people are not immune. They perhaps are a little more cavalier than the rest of us in terms of really following the social distancing policies that have been put forward by the advisory committee to the president about this pandemic. And of course, all you have to do is look at spring break and all the kids that are down on the beaches in Florida. Also, over the weekend, a lot of St. Patrick's celebrations and bars and things like that in the city and elsewhere. Young people are not immune, and certainly risk the possibility of getting pretty sick. They're perhaps not dying, but still getting pretty sick. And of course, then bringing home that infection to their parents and their grandparents and other elderly individuals who they may come connected to. The other thing that I think is important, a study was published earlier this week on Tuesday in the New England Journal of Medicine, which had to do with the relative stability of the virus, aerosol, which is really how we contract it, and on different surfaces and things. And I wouldn't say that the data that was published in that paper was very different than what I would have expected from a typical respiratory virus, but again, it shows that the virus is viable, for at least 3 hours in aerosol. So if somebody sneezes and those aerosol droplets stay in the air. If you breathe those in, that you can get an infection, up to 3 hours after that person released those. And, again, they looked at different surfaces. They looked at cardboard. They looked at, different metal surfaces, things of that nature, and found that, the virus hangs around for several days. And that's, again, why we have to be so careful about, washing your hands and not touching your face and touching your nose and eyes and things like that, because, that virus could have been deposited there yesterday. It's still there and it's still viable. So I think that's, in my mind anyway, those are sort of the two significant, let's say, news items about this pandemic that have come out this week. So.
Thanks. Ken, one of the things I'll mention is that you also, your role, one of your other roles at the college is the chair of the Infectious Disease Response Committee. And you've been advising leadership as we've learned about this virus. You've been in conversation with Sister Carol, providing her with your expertise and perspectives on COVID-19 and everything we're learning about it. What do you see any unique characteristics of the spread of this virus, and anything? Obviously, there's so much unique about this in terms of our national and global response to this event compared to prior events. What can you tell us about that I know you've been telling? Sharing on other
sure. First off, I want to add a disclaimer, which is, I think that everyone, the most learned of scientists, the most respected of infectious disease physicians and virologists, have to, all of us agree that this, in many cases, is uncharted waters. And so we're sort of making educated guesses when we try and predict what's going to go on or explain what's going on. So with that sort of disclaimer, I can one factual piece of information, which isn't, a projection or anything, which is actually real, is this particular virus spreads much more rapidly among humans than previous coronaviruses, and in particular SARS, which was a coronavirus that arose in 2003 in China as well, had also the unique characteristic of being fatal. Most coronaviruses are not. Most coronaviruses just cause common colds, usually during the winter. But as a result, this virus is about 10 times more infectious than SARS. And one reason, perhaps, is that the way viruses work is that it's a very specific interaction between the virus and the host cell that they infect. And the way this specificity is dictated is that there's a molecule on the virus and a molecule on the host cell. And the two of them come together. It's almost like a lock and key kind of situation. And in this particular version of the coronavirus, it actually recognizes a molecule on the surface of human cells, which is called the angiotensin converting enzyme 2. And it, again, binds to this. There's a spike protein on the outside of the virus that binds to that ACE2 receptor and binds at a much stronger affinity. So it's a much tighter bind. A lot of times with viruses, it's sort of the kind of thing that, obviously the two molecules have to come in contact, but it's not always a permanent contact. So, they'll come in contact and they dissociate. They associate, they dissociate. In this case, when they come in contact and associate, they stay bound. And that's at least the guess that explains why these viruses seem to be more infectious for humans than in the past. And of course, the other thing that's unique is this whole idea of social distancing. There's been a lot of other pandemics over the course of centuries, really, and I don't think there certainly have been numerous examples where quarantines have been imposed. And perhaps it has a reflection to do with our society these days. But the term social distancing, of course, has been really coined in response to this pandemic. But it's certainly something that is an interesting strategy, in that viruses need susceptible hosts. It makes a lot of sense because if the virus no longer has any susceptible hosts, then it will die. Then it will basically disappear because it can't exist without having other hosts to infect. And so the idea is really to try and reduce the number of individuals that the virus comes in contact with. The hope that eventually it no longer can infect because there won't be anything else for it to infect. So I guess those to me it would be the most unique things about the fact that you have this virus that has this extremely high affinity for the particular cells that it's specifically infects, and then the fact that you have this idea of social distancing as a way of trying to reduce the number of individuals overall that are infected. Okay.
One of the interesting things I think we're seeing in sort of the broader media is it's I think and we all just individually are struggling with. It's very difficult to think about this phenomenon as we currently experience, and to try to compare it to something else.
Mm-hmm.
And from your perspective, does anything compare to what we're seeing now in terms of what in the research? Other kinds of viruses, pathogens, anything else in terms of the response of the scientific community, the public health community, that helps us place this in some kind of context.
Right. Well, again, epidemics have been around, pandemics have been around for literally centuries, as I've said, and certainly even in recent times, we can go back to 1918, which was the devastating flu. That was the Spanish flu, that killed 50 million people. We can go back, you don't have to go back that far. You can go back to 1957. You have the Hong Kong flu then, that killed about 1 million. You have the AIDS epidemic, in the 1980s. And you have SARS, as I said before, in 2003. What has been clear, the numbers of fatalities of these epidemics has dropped precipitously with time, so that the ones that were, literally, hundreds of years ago, had millions and, 30, 40, 50, even 100 million. Smallpox, for example, certain types of, fever and the plague and things of that nature. So what has happened, and you raised an important point, is that certainly science has progressed tremendously, and what's been clear, if we look at SARS in 2003, if we look at AIDS and HIV in the 80s, is that when something like this happens, and this is even true with COVID-19, immediately you mobilize the scientific community, the research community, and immediately people start working on it and make remarkable progress, really, in terms of time. This virus was barely known, and a sequence was already available by the end of last year. Sequence of the genome of the virus, which, again, a lot of information can be learned from that. And that serves then as the basis for then trying to develop antivirals, develop a diagnostic test, develop a vaccine. So I think that we have seen similar kinds of things before, and it's pretty remarkable how quickly the research community, reacts and responds. And then, what has happened too is there has been a desire on a part of the scientific community to share information. And so, for example, if you go to the scientific journals, normally you need to have a subscription, obviously, to the journal to actually be able to read the papers. But there is a policy that permeates the entire scientific literature that anything about coronaviruses is open access. And so even if you don't have that subscription to New England Journal of Medicine or to Nature or to Science, if it's a paper about coronavirus you want to read, you can click on it and have instant access to it without having to register or pay anything or anything else like that. And again, the whole idea here is to try and have sharing of data and sharing of information among the scientific community so that we're not rediscovering the wheel. You don't want to have to do the same thing that another lab has already done. There's also accelerated reviews of those papers, and so again, things are being published very quickly, and again, that's important too. So although it may sound like it seems to be taking forever to come up with some kind of a treatment or some kind of a vaccine, in actuality, in terms of the normal calendar that, the normal amount of time that it would take to do these kinds of things, in times of crisis like this, the scientific community really accelerates their work. And so I'd have to say, I'm optimistic that I understand, I think this week that they already started some preliminary vaccine trials. Again, that doesn't mean that there's going to be a vaccine available. Anthony Fauci, discussed that last week and said, just the sheer process of going through the trial periods, will take at least a year probably. But again, developing a vaccine that quickly is still pretty remarkable. So I think anyway, those are the major things that are, again, that we're seeing that is of relevance in terms of the response of the, and of course, the epidemiologists have, been out there talking about, flattening the curve and things of that nature, which again, the public health group is also very active as well. And, again, because of all the numbers of epidemics and pandemics that we've had, the public health people have a very good idea of exactly what to expect. This isn't, anything unique in that sense. It's doing exactly what you would expect to see with any kind of a infection that's become a worldwide problem. So.
Yeah, thanks. And you mentioned a couple of things about this from my layperson's perspective, what I'll call the mechanisms of this particular virus. And I'm just going to jump ahead in terms of the couple of questions I had for you today, and sort of, you've referred to the fact that the coronaviruses are common, for lack of a better term, seen coronavirus. They circulate every year. Again, just from a layperson's perspective, I'll describe it this way. Is there anything else you could describe about the mechanisms associated with this virus and what makes COVID-19 unique as a virus from a researcher's perspective, and I think in ways that can inform our community in deeper ways than we might be getting from the fleeting media reports we're hearing right now?
Right. Well, it is, a fairly common respiratory virus. And what does it mean to be a respiratory virus? Basically, a respiratory virus is a virus that enters the body through the respiratory system and remains localized and actually infects cells of your respiratory system. So it's going to infect the cells of, your lungs, your nasal tract, things of that nature, but it doesn't go anywhere else. So it's sort of self-contained, and it's actually a relatively small, what we call incubation period. So from the time you get exposed to the virus to the time you start to feel symptoms, is usually 3 to 5 days, something like that. Other viruses that have to travel, enter bloodstream and then get taken to other parts of the body, there the incubation period could be as much as 14 days or even 21 days. But respiratory viruses stay pretty much in the same place that they enter. They're unique in that they actually have an optimal temperature, which is a few degrees lower than other viruses, because your respiratory tract is actually a couple degrees cooler than the rest of your body. So in Celsius, our body temperature is 37, but the temperature of our respiratory tract is actually 35. And so these viruses actually turn out to be heat sensitive. And that's one aspect that has been talked about is, will the virus begin to, the incidence of infections begin to drop as the weather gets warmer and more humid. And there's certainly people who believe that, that will be a significant part of the control of this viruses. And again, SARS did this exact same thing. Come in the middle of April, SARS really disappeared. It pretty much was substantially reduced in numbers of infections that were reported come the middle of April. So we can hope that that's going to be the case here too. The other thing that is important is that this is an RNA virus, and all these coronaviruses are RNA viruses. This means that the genome is RNA. And when the genome replicates, it replicates by an enzyme called RNA polymerase. And this is an enzyme that doesn't have any kind of proofreading. So sometimes, and RNA viruses tend to replicate very rapidly. And so sometimes, like anything that you do rapidly, sometimes it's sloppy. Sometimes it's not exactly perfect. And so it inserts bases, the wrong base sometimes. And as a result, you get mutations. And as a result of those mutations, you then have viruses which have unique characteristics, some of which are disadvantaged and they actually die off because they can't survive, but some of which, depending on the environmental circumstances, may in fact give them a selective advantage. So it's sort of survival of the fittest, and you have let's say an array of different viruses with an array of different genes. Some of the genome obviously has to be stable and the same, but there are some places where, in fact, there can be some variation, and depending on the environmental insults that the virus is exposed to, some of those mutations allow it to survive better than others. Now, this particular COVID-19 has a genome that is about 90% identical to SARS. And those viruses are actually have some unique characteristics that are different from the other coronaviruses, the coronaviruses that are common, that cause those winter viruses and that are not fatal at all, but just annoying. And one of the things that's unique is they have 13 open reading frames. An open reading frame is basically a stretch of a genome encoding a protein. They don't know what the protein is, but those, in this particular case, but those proteins, some of those could have to do with virulence, for example. And it could be, the reason why SARS and COVID-19 are more fatal than the other coronaviruses is by virtue of whatever those open reading frames are and what they encode and what those then subsequently, the effects they have on the body. So, what we have here then is the sort of the core of the virus is common. We see it all the time. And again, it has all the same characteristics that other respiratory viruses have. But then, as a result of this sort of sloppy replication, there's the possibility of some unique mutations that arise that can certainly influence then the range of individuals that are infected, the severity of infection, all those kinds of things. And the point I had made earlier about the fact that that this virus seems to bind the receptor in the human cells stronger is another example of that. So that the sequence of the spike protein has some unique bases in it that encode some unique sections of that protein that are responsible for that some strong binding. And so now that being said, I will also say that those unique sequences also make very good targets for potential therapy. And so it's sort of a double-edged sword. On the one hand, it's responsible for this virus being unique and difficult as it is at this point, but that will also serve to be its sort of Achilles tendon, in the sense that that's what scientists are trying to target. And if they can inactivate that somehow or other, or block it, or whatever it is, that will prevent the virus from being infected. So I think that those, that if we're talking about, commonality versus uniqueness, those are those 13 unique reading frames that are encoding, certain factors, which probably are responsible for the fatality of the virus. But they also should be able to serve as therapeutic targets as well.
Thank you. It is both concerning and encouraging to hear about this from that perspective. One of the other things that I know you've been speaking about as you've been advising the college is the data that we're seeing, the way that they're being presented. And I think obviously this phenomenon we're seeing from an empirical perspective, flattening the curve. We're seeing a lot of scenarios in which if social distancing is successful, that would have the net effect of flattening, but then perhaps extending the curve. Thoughts that you have about those things that we're seeing from a public health perspective, just in terms of the data, the way that they're presented.
Mm-hmm.
What are they telling us?
Right. Well, I think sometimes this, daily posting of new cases can be, disturbing in the sense that, we're doing all this sacrifice, we're staying in the house, we're social distancing, we're, staying together, trying to reduce the amount of interaction we're having with individuals, but yet, the numbers keep climbing. Well, the numbers are going to climb as you do more and more tests, and so in that sense, the reporting probably is doing a disservice, because it may be indicating to the general public that, all this stuff isn't working. We keep getting, more and more cases. But in actuality, we can't really tell whether social distancing is controlling the infection at this point, because we're in the middle of it. You don't know what the numbers would be if you didn't social distance, and so, if you look at the numbers in terms of what happened in China, it took anywhere from, 8 to 10 weeks between when those the numbers of cases were climbing exponentially, and then when it flattened out. And it's flattened out, and actually, I think today or may have been the first day that they reported a reduction in numbers. And so I think, people tend to feel at this point that the situation in China has already peaked and it's on its way down. But you can't really know the answer. I think we may have jumped a little bit further ahead at the beginning than China. China actually was very slow in responding. And so it may have allowed the virus to get, or the infections to get out of hand, in the sense of many people were infected before there was any attempt to respond and do anything. Here, I'd like to think that we were pretty proactive about it, and even going back to December and January, people were being held, for 14 days, coming from China or coming from Asia. And so they tried to sort of reduce the number of individuals, coming in contact with the population here in this country. Certainly Anthony Fauci was very proactive about pushing forward this idea of reducing gatherings of significant numbers, things like that. Really pushing forward the whole idea of social distancing fairly early in terms of our numbers. So I hope that we will see a significant flattening of the curve. But I guess the concerning thing is that people will get discouraged, because before we see that evidence that it's flattened, we're going to see increases in numbers. There's no question. And, they're talking about, 1 million or 1.5 million tests being available by the end of this week, and 4 or 5 million available by next week. The more tests you do, the more positives you're going to get. And so that still should be put in perspective, though, in the sense of, as I say, you don't know how many you would be getting if you weren't, being careful, and if you hadn't imposed some of these policies in place. So I think that's, I guess that's the only, at this point, the only thing I can say. I think, again, one has to be careful of the sources of the information that you're getting. I would place tremendous trust in CDC. This is their business. They have professionals, and this is not their first rodeo. They've been around. Again, all those epidemics we talked about in terms of flu, in terms of HIV, in terms of SARS. They were right in the middle of, and so that's what their specialty is. And so any of those reports coming out of there, any of the numbers that are coming out of there are vetted and are certainly valid, and their recommendations based on those numbers, I think, again, are valid. So I think that the news agencies that are using CDC data or WHO data, if you're talking about worldwide, are certainly, can be trusted, and those numbers, I think, can be trusted. But otherwise, you certainly don't want to depend on anecdotal information about, 5 cases that were in Conshohocken or something like that, but that some neighbors are talking about.
Thanks. I mean, it's just been a remarkable thing to watch. I want to remind everyone against when we're having this conversation, because I really appreciate you doing this, and I hope we'll be able to have a couple more of these, just to support all the work of our community as we do this remotely. But here we are on March 19th, and looking at the numbers as of today, they're going to change. And
Mm-hmm.
so, I just think it's good to set the context for, again, when we're having this conversation.
Yes.
Yeah. I wanted to return to an earlier question I had for you, and that was sort of what can we expect as organizations moving through this? What are you seeing in terms of these recommendations for social distancing? And not to ask you to, again, sort of as a disclaimer, we're sort of in such new space here, and not to make specific predictions, but just what do we look at when we see these windows of time? And as I see it, there's some 15-day windows of time that are being presented to us.
Mm-hmm.
It's 45-day windows of time. A cycle of 180 days, a year and a half, when we would actually go through a cycle of developing a vaccine. Can you give us a sense of these time frames, what they mean, and what we should be looking for as organizations? We're so anxious about reconvening as a college, but we just don't know how long it's going to be until
That's right. Exactly.
it
Yeah.
happens. And just anything you can say around that as we look to kind of, as organizations in general, try to make these decisions.
Yeah. Yeah. Well, again, I think unfortunately, if we're going to make the decisions on the basis of continued number of cases reported, I would predict that those numbers aren't going to go down in 2 weeks. So a lot of places have made decisions about closing till the end of the month, until April 1st or something like that. I think that, as I said before, the more tests you do, the more positives you're going to get. Now, it's all relative in the sense of, well, the more important thing is how many people are dying, and is the healthcare system being overwhelmed? I mean, one of the Fauci's major point about social distancing was not so much. I mean, obviously it was to try to reduce the number of infections, but also the reason why that was so important to not only reduce it but slow it down is to give us time to respond. The hospitals and the clinics and the health care facilities, needed time to prepare, needed time to get sufficient supplies, needed time to train the healthcare personnel. And so if you could slow the process, as you said, not only is the curve flattened, but it's also extended, so that, you don't have this huge influx of thousands of people overwhelming the hospitals and the health providing facilities. And so in terms of trying to figure out what's going on, whether it be 2 weeks or a month or 3 months or whatever it is, I think it's going to be hard. And I don't really have any crystal ball. As I say, I think as we get into the spring, I really do believe that the virus will be less stable and not as easily infectious, in the warmer weather, the more humid weather. If you sort of look at, the Southern Hemisphere, where, they're toward the end of their summer, there have been very few cases in places like South America, for example, so that indicates, that perhaps this is a virus like all the other respiratory viruses that tend to be very, sensitive to the heat. And let's hope that's the case, and looks like this year we're going to have a slightly warmer spring, earlier spring, and perhaps an earlier summer, so that may be in our favor. But I really do believe, as I say, the numbers are going to continue to climb for a while. If I guess the question is, what criteria do we use? We as an institution, for example, in terms of having our students come back and reinstituting classroom teaching, the city and state governments relaxing the rules about what restaurants being able to serve people in the restaurant itself, stores opening, malls opening, things like that. I don't know what criteria other than numbers, unfortunately, that you can use. If that's the case, then we probably are probably, we can hope that by Easter or by the end of April, where we start to clearly see indications of that drop. Again, if you look at China, China was 8 to 10 weeks. So, if we count, let's say, the first week in March as our first week, then, by the end of April, that's 8 weeks. And maybe, we'll start to see a drop then. And if I'm right that we, sort of got a head start on it, and we didn't allow things to get out of hand quite as much as China did, maybe less than that period of time. But again, I have no crystal ball, and it's, I don't even know if it's an educated guess, to be honest. Maybe it's more hopeful than anything else.
Yeah, there we go. It at least gives us a sense of the criteria that folks are using in making these measurements. Just before going to a couple last questions, I know you shared your perspectives on Dr. Fauci, Anthony Fauci, and your familiarity with his work. I wonder if you could just say a couple things about your perspectives on his work as head of NIAID and what the implications are there.
Yeah, well, Tony Fauci's been the director of the Institute of Allergy and Infectious Disease at NIH for almost 40 years, or actually over 40 years, I think. And he is extremely highly respected, extremely accomplished in terms of his, expertise in infectious diseases. Has been an advisor to 4 different presidents and is completely apolitical in the sense that he's advised Republicans and Democrats, tells it like it is. And I think that's why he's been so successful, is that, he's very straightforward. I think that's been clear in his interviews and comments. He was probably the first person who really contradicted Trump in what Trump was saying versus what reality was, and he was very clear about that. I think he's extremely talented in that he can explain extremely complicated things in layman's terms, again, being straightforward, but not dumbing it down either. I mean, making it very clear, the complexity of what's going on and the science behind what's going on. But his experience is just so phenomenal because he was there for the AIDS epidemic. He was there for SARS. He's there for Ebola. He's there for West Nile. He's there for, Zika. He's not at all phased. He's very level-headed, doesn't get, overly excited about these kinds of things. He approaches the problem the way it should be approached in a scientific fashion. Let's figure out what the problem is, let's figure out what the best solution is, and let's go ahead and do it. I think we're in very good hands there, and I'm happy that the president included him in his advisory group because of all the people that are in that advisory group, he probably has the most experience in dealing with the problem. So. On an aside, he and I are both Holy Cross graduates, so I'm a little bit prejudiced about him. But again, you don't survive as a director of NIH for 40 years if you don't really know what you're doing. It's probably the bottom line.
So I'm switching our conversation in the last couple of moments to the Chestnut Hill community and the moment we find ourselves in as we've moved to this sort of distributed online instructional model, and know that hopefully a lot of our students and our faculty will be seeing this conversation, other conversations. How about your teaching right now? How's it changed in terms of this moment that we find ourselves in?
Well, it's been a challenge, I'll say that much. I certainly, in what you'd have to label as a traditional professor, in the sense that, in my courses, for the most part, they're lecture-based, significant number of PowerPoints, and I usually stand up there and walk people through the PowerPoints. I'm certainly open for questions and such, but there's not a lot of discussion. You're teaching virology or molecular biology or cell biology. It's a very fact-based course. And then, where you have some discussion is when you're trying to solve problems and things like that. I always have there's a significant number of PowerPoints for each of my lectures. And so the challenge was then, I didn't want to just simply say, okay, well, review the PowerPoints, and if you have any questions, email me. And so I've used actually Canvas Studio to do voiceovers of those PowerPoints. So in essence, I give the lecture and can record it. They don't see me, but they see the PowerPoints. And of course, I can actually point out on the PowerPoints using the mouse, computer mouse, the specific things that I want to emphasize. And then what I've done is I've incorporated additional videos, which, again, in some cases provide animations or, other explanations. And I've written study questions, which, I've asked them to answer as they go along and then send them to me afterward, which gives me an indication, first off, that they actually listened to the lecture, but also the study questions in some cases are a little more open-ended. They're not, some of them are fact-based, and I emphasize that I want them to use their own words in answering them and not cutting and pasting off of my PowerPoints. But some of them are also more essay-based. So again, I have some clear idea of whether they understand what they're doing or what they're learning. So that, having to learn that technology and having to, make certain that the recording works well, and things like, well, it's better to do them in 2 or 3 segments of 20 minutes rather than doing a 75-minute single recording, because, well, it takes forever for a 75-minute recording to download, for example, whereas if you do it in 3 or 4 segments, then it's going much faster. So I'm learning as we go along, and it's a challenge. And I have to say that when I get it right, I feel very good about it. I feel like I've actually accomplished something. Let's hope that continues. The challenge I have is I also teach senior seminar, and I have 27 students in there, and that's a completely different type of course. And normally they have to have a research project of some sort, because we managed to get through spring break, the challenge, I had, from the very beginning of the course, I told them that they had to pretty much have their research project completed by spring break. So then the question is, okay, they have to take that and turn it into a poster and turn it into a formal PowerPoint presentation and then a scientific paper. And there I've been working one-on-one with them. Their posters are due now, and we've been trying to sort of exchange rough drafts of those posters. The problem there again is motivation. They have to be, motivated, and they have to. It's always very easy to procrastinate on those kinds of things, and so I have to try and get them to get me the drafts, and we can have an exchange back and forth. And then once we finish that, which hopefully will be done by the end of the month, then we have to move on to the PowerPoints for the presentation. Now, whether they ever will show these posters and whether they will ever give the presentations is another issue, but I think it's still worthwhile doing it. And I've always told them, even in class, even when we do all this stuff, that the advantage of having it is that when you go for an interview, you have something to talk about. You can actually show, you can take a print of your poster, and you can say, this is the poster I gave for my capstone project for my major, and this is what I studied, and this is what I found out about it, and you'll have something that you can start a conversation about. So I think it's still, even if we don't wind up going back and we don't have senior seminar poster day and we don't have the formal presentations, it's still worth doing. As an experience as a scientist, they have to learn how to do that. And they still can use it. So, but that senior seminar is certainly not something that's very easy to do online, obviously. But we'll, do the best we can.
Yeah, and I appreciate you sharing that, Ken, because I do think it's evocative of so much of the work that faculty do with students, particularly in those seminar experiences. And for those who may not know the college, or just to remind those of us who do, that experience of the whole college community coming together, at the end of each semester, where students present their seminar projects, literally the whole of the college community comes out, faculty, staff, administration, members of the community. And, we're a week into this experience. And I think one of the things that we're anticipating already is how can we do everything we can to recreate all those important interactive components of the learning experience at the college, which are so definitive for us, place-based. And I think that's one of the reasons why so many of us are anxious to get back. I mean,
exactly.
That interaction defines the learning experience. So I mentioned our students, and Ken, I'm so grateful for this conversation. One last question before we end our time. What should Chestnut Hill College students be thinking about this moment? What should we be teaching our students about this moment that we find ourselves in right now?
Well, I think that we're bombarded by information. You turn the TV on and that's all you hear. You check your phone and you're getting reports every 5 minutes about, different news conferences or different, groups talking. We're on social media all the time. And so certainly the first thing I think that we have to make certain our students know, and this is true, it would be true even if we weren't in the middle of a pandemic, is, what sources of information can you trust? And I think it's particularly important when you have a scientific problem like this, because let's be honest, society in general are not as comfortable talking about science as they are about music or art or some other kinds of areas or topics. And so, it's dangerous that you might very well hear things which are not true. They're not scientifically based, that you might find in Facebook or Instagram or other kinds of social media means of communication. And so, we have to, make certain that they understand, what's the source of the information that they're getting. And again, something like this, CDC by far is the, most important source of information. WHO, another good source, certainly anything that comes out of the NIH as well. But I fear that people, will just sort of look at the headlines and not pay attention to the source, and then repeat that information, to others as fact when it isn't fact. So I think that's certainly one thing that they should be, aware of, and we should be teaching them. The other, I think, again, sort of is a reflection of today's news, and that is, they are young, but they're not invincible, even though they think they are, and that they, viruses don't necessarily discriminate. They don't. Yes, a lot of times elderly people are more adversely affected by infections than young people, but young people still get infected. They can, still suffer and experience pain. And so they shouldn't be very cavalier when it comes to an infectious disease. It's important that they realize, and I think they do. Certainly, the couple of days that we had on campus before we left, I was actually very pleased and impressed that some of the discussions that I did have, young people, raising the issue that, I know I probably won't get it or may not get it seriously, but, I live with my grandparents, or I see my grandparents every week, and I certainly don't want to be the conduit of bringing them a potentially fatal infection. And so they're aware of the fact, and we want to make sure that they continue to be aware of the fact that, they can be vectors of infectious diseases, and could very well, even if they are not experiencing really debilitating symptoms, they could be the source of infection for others who, could. So I think those two things to me are the proactive in trying to make certain our students are aware of those two things. That be careful of your source of information, especially when it comes to science. And secondly, be careful of not taking these kinds of problems, these kinds of infections seriously. That's it.
Ken, I'm grateful for your time. Many of us have been the beneficiaries of your expertise over the past couple of weeks, and I'm so pleased that you've taken the time that we can share this with the broader college community, with our students, and with others as well. Dr. Kenneth Soprano, professor of biology, also chair of the Infectious Disease Response Committee, and former vice president for academic affairs and dean of the faculty at Chestnut Hill College. I've been grateful for your time, and thank you, and wish you well as we move through the next few weeks or months of this experience.
Thank you, Chris. Appreciate the opportunity.
Absolutely. Yeah, you too, Ken. Thank you.